different, and so that's why we need all the structures to support the body functions. If you look at the structure, you can see that the spinal cord is basically surrounded by this meninges, these three level of they are basically structures with different functions, in general protective functions that surround the spinal cord and are located between the spinal cord and the vertebral column. Here you can see in detail the structure of meninges. So keep in mind that we have meninges that surround both brain and spinal cord. This is an example. We have the brain, but it's the same situation for the spinal cord. So you have the brain tissues in here. That the first layer is the pia mater. It's a really thin structure. It's a really thin membrane, and is important for the blood supply to the brain using the arteries. Then this pia mater is basically separated by the arachnoid, the subarachnoid space, and then we have the arachnoid membrane. This kind of empty portion organized with this structure is the subarachnoid space. Subarachnoid space that we are going to see is important for the cerebrospinal fluid location and movement. Basically, the subarachnoid space is where the cerebrospinal fluid is located, and it has this protective function for the pia mater and the brain located down the pia mater. Then moving through the meninges, we have, as we said, the arachnoid membrane, and then this portion that is the dura mater, that is the thickest portion, the thickest membrane. You can see that the dura mater is strictly connected with this cavity, these structures that are the sinus. Sinus are these structures, they are not empty, they are not composed by tissues, but they are associated with veins, and so they support the drain of blood from the brain. So it's the way through which the brain the blood moves out of the brain using this sinus. And as you can see, there are also this kind of portion that comes from the subarachnoid space that moves into the sinus. We have these structures, called villi, because they support the exchange of ions, vitamins from blood and the cerebrospinal fluid that basically moves, is located into the subarachnoid space. That's why all the structures are connected by each other in order to allow exchange of ions, exchange of vitamins, but of course we can also have the exchange, the movement of some harmful molecules, substances, pathogens. That's why our brain must have an increased level of protection compared to the other organs. And so this protection is a function of meninges. We also have the cerebrospinal fluid, but an important protective barrier is the blood-brain barrier that avoid this kind of exchange. It protects the brain from any kind of harmful molecules that comes from the blood, from the capillaries. As we said, of course, we also have this extracellular space that is filled with fluids. Two different fluids, the interstitial fluid and the cerebrospinal fluid. The cerebrospinal fluid is located in between the pia mater and the arachnoid membrane, is in this subarachnoid space. For what concerns the interstitial fluid, it is located inside the pia mater. So all the meninges, all these structures are filled with fluid. In this way, they can support the brain movement, the brain rearrangement, and avoid any kind of trauma that can come from outside or that can come if the brain was strictly in contact with cranium. So that's why we have this level of membranes. Also, the cerebrospinal fluid is now considered really important as a biomarker for neurodegenerative disease. So in the diagnosis of some pathologies, because it is considered a kind of representation of the brain situation, of the brain health situation. So if there is something wrong, if kind of unfolded protein, misregulated protein associated with disease like beta amyloid for Alzheimer's disease, they can also be visible into the cerebrospinal fluid. So it is really important for the diagnosis, but also as a biomarker, thanks to some specific component that can be proteins. We are identifying some microRNA into the cerebrospinal fluid that are a kind of realistic representation of the brain situation and can also be used for the diagnosis of pathologies, of the stage of some pathologies, because the cerebrospinal fluid is strictly in contact with the brain. It receives molecules, factors, soluble factors that comes from the brain. So it's useful for the diagnosis, and if we want to develop a specific therapy for this neurodegenerative disease. Also in the cerebrospinal fluid, we can find extracellular vesicles that transport lipids, RNA, microRNA, as a way to send toxic factors, as a way to also take back important components to support the neurodegeneration. If you look at the cerebrospinal fluid, as we said, it is located inside the meninges, and in that kind of extracellular space. For the region where we can find the cerebrospinal fluid, you can observe the presence in the brain of ventricles. We said that ventricles originated from the lumen of the nervous system. When we talked about the development of the nervous system, we said that it was a hollow structure, a hollow tube, and from the lumen, from this end cavity, we are going to have the formation of ventricles. So in the brain, we have two lateral ventricles, the first and the second ventricles. They, as you can see, connect to the third ventricle, that is this portion in here, through this opening. Then the third ventricle moves down here into the fourth ventricle, into the brain stem, and then the fourth ventricle in here continues into the spinal cord through the central canal. So these are the region where the cerebrospinal fluid can be found in ventricles and in the space, as we said, between the pia mater and the arachnoid membrane, into the subarachnoid space. These structures are filled with cerebrospinal fluid. So we have two lateral ventricles, a third ventricle that connect the two lateral ventricles, and then the fourth ventricles into the brain, the central canal that move inside the spinal cord. Why is the cerebrospinal fluid important, and which is its origins? So this is a view of our brain, sagittal view of our brain. As you can see, this structure, the main structure, is composed by the cerebrum, and then we can see the ventricles. This is the lateral ventricle. Down here we have the fourth ventricle, and the cerebrospinal fluid comes from the choroid plexus. The choroid plexus is this red structure that surrounds ventricles in here and down here, close to the third ventricles. Talking about the composition, it is similar to plasma. Why? Because ions, vitamins, molecules, nutrients comes from the capillary. So basically this is the ventricle. The choroid plexus, this structure, surround the ventricle and also surround the capillaries that lie close to the ventricles. Thanks to this localization, you can say that thanks for the osmotic gradient, ions, molecules, vitamins moves from the capillary to the cerebrospinal fluid, creating its composition. That's why the composition of the cerebrospinal fluid is similar to plasma, because the majority of these molecules comes from the blood and is located basically down here. This is the representation of the third ventricle, the choroid plexus that surrounds the third ventricle. It's the same for the choroid plexus close to the fourth ventricle. So basically, once we have the formation of the cerebrospinal fluid, the composition of the cerebrospinal fluid, it is going to be localized in this portion if we can see the representation is the green portion, the subarachnoid space. As you can see, it surrounds the spinal cord and the brain. So this is the brain, the cerebrum. Then close to the cerebrum, you can see the pia mater, this thin layer that surrounds the cerebrum. And then you can see the green portion that is the subarachnoid space where the cerebrospinal fluid is located. And then once again we have the arachnoid space and the dura mater, that is the external layer of the meninges. These structures you can see on the top here are the arachnoid villi. I should have, yes. So this is the structure. This is the subarachnoid space that was the green structure in the previous picture, where the cerebrospinal fluid is located. And then these are the villi. As you can see, villi are connected to the sinus. The sinus were that kind of region that were responsible for the blood drain from the brain. And it's through this structure, through this villi located into the sinus, that cerebrospinal fluid can be reabsorbed. So the cerebrospinal fluid is produced into the choroid plexus, and it takes ions, vitamins, molecules from the brain, from the blood. On the opposite way, using this structure, this villi that are going to be located into the venous sinus that receive the blood from brain, we also have the reabsorption of the cerebrospinal fluid. So in most, it is localized around all the brain structure. This is the cerebral cortex, the pia mater, the subarachnoid space where the cerebrospinal fluid is located, then this structure, the arachnoid membrane, and then the dura mater, this inner layer that is in contact with the sinus, where the villi are going to be located for the reabsorption of cerebrospinal fluid into the blood. At this point, so this is the main composition of the structure we have in the brain. So the meninges, the cerebrospinal fluid, the cranium. Yes, this is just to summarize the composition of the cerebrospinal fluid. So basically, we said that it's really similar to plasma. The sodium concentration is similar to what we have in the blood. The only difference is related to potassium, that is a little bit lower into the cerebrospinal fluid, and hydrogenous, that is higher. But more or less, the structure, the composition is the same. As we said, it is important because it represents the chemical environment in the brain. So for clinician, in case of diagnosis of neurodegenerative pathologies, the cerebrospinal fluid composition is really important. It is getting important in these last years for this diagnosis. We also have the other protective structure, that is the blood-brain barrier. What is this structure? So basically, it's a structure that we find only in the nervous system. It is not present in the other organs in our body. It's another protective layer. It's a barrier, and it is permeable, but with a high selective permeability. So basically, it avoids that harmful substances, pathogens, bacteria that are moving into the blood arrive into the brain. So this is another layer that we have close to capillaries. Here you can see the structure of the blood-brain barrier. So this is a capillary. This protective barrier is composed by pericytes and astrocytes. Thanks to gap junction that can be that are present in between these cells, so you can see the pericyte localization strictly connected to the capillary and surrounded by the astrocytes, the food processes of astrocytes. The presence of gap junction, these closed structures avoid that molecules, pathogens, bacteria move from the blood to the brain, because usually in the capillaries, the cell-to-cell junction, the presence of this leaky cell-to-cell junction promotes the free exchange of molecules, soluble factors. In this way, in the brain, we want to avoid this situation because it would be easy to damage the brain. It's a really delicate structure. So we need another level of protection that is supported by the presence of the blood-brain barrier. That's why we have this structure. And thanks to molecules, paracrine molecules that are secreted by pericytes and astrocytes that are recruited at that sites, where we have the formation of these other protective layer in the brain. So yeah, it protects from toxic, any kind of molecules that are water soluble, and so they can easily move from the blood to the brain. For example, the dopamine. So the brain-blood barrier is not permeable to dopamine. The problem is when people affected by Parkinson's disease have a reduction in the dopamine, because dopaminergic neurons are damaged. So the dopamine, that is this neurotransmitter, neurohormones that modulate the transmission in the dopaminergic neuron function, is compromised. So we have a reduction into the dopamine. The most common therapy for Parkinson is the administration of L-dopa, that is a precursor of dopamine, because our brain, the blood-brain barrier, is not permeable to dopamine. So the L-dopa can be transported across the blood-brain barrier using some specific transporter. Something similar happens also for antihistamines drugs. All antihistamine drugs were able to cross the blood-brain barrier, and so activated some kind of centers in our brain, alarm center in the brain, that as side effect give us the side effect of the antihistamine. So the sleepy situation, headache, that does not happen for, let's say, modern antihistamines, because they are not permeable to the blood-brain barrier. So our brain is protected from side effects that can come from these molecules. That's why we need this support, these and other barrier structures. We can have a break, and then we can quickly introduce the spinal cord, and next time we can talk about the brain. It would be better to have a break now. We can have a few minutes. Okay, guys, so we okay. Okay, we can go ahead talking about what are the metabolic requirements of the brain. It basically needs oxygen and glucose to produce ATP as an energy support. Glucose is the most important support for the energetic requirement of the brain, and it basically comes from it is half of the glucose that our brain needs comes straight away from the brain. Brain is responsible for half of brain consumption of glucose in our body. It is important because if there is something wrong with the glucose amount in our body, like in case of hypoglycemia, so we have a reduction in glucose, since the glucose is an important source of energy in our brain, it's easy to understand that as a consequence we can have kind of confusion, unconsciousness, in the most severe condition, even that, because of course there is a reduction in the amount of glucose, so reduction in the capacity of the brain to support all its metabolic requirement. Another important origin of energy, I mean, ATP also comes from oxygen. Oxygen is can easily cross the blood-brain barrier. It does not need any kind of specific transport. It can cross the barrier and move straight away into the brain. It is an important energy source for our blood, for our body. And in case of oxygen loss, in case of the blood flow in our brain is interrupted, because keep in mind that oxygen is transported by blood using the hemoglobin in the blood. So it means that if there is any kind of interruption into the blood flow into the brain, and so oxygen is not arriving into the brain, as a consequence, we can have some kind of severe situation. So basically, people can lose consciousness, or they can have severe damage into the brain, even if the oxygen is not coming for a few seconds, because of course they are not receiving the energetic support. The brain is not receiving its energetic support, and so the brain functions are impaired. That's why it is important to regulate the amount of oxygen flow, so the amount of blood flow into the brain, as well as the glucose concentration must be kept in a kind of normal physiological level. Otherwise, if we have an increase in the glucose or a decrease like the hypoglycemia, brain can be severely damaged. So we can have really severe consequences if the metabolic requirements of the brain are not supported, and they are mainly related to oxygen and glucose support. So I want to quick introduce the spinal cord, talking about the central nervous system. We said that the central nervous system is composed by two main structures. One is the brain and the other one is the spinal cord. Spinal cord is all these components that move down in our body, protected by the vertebral column that is organized into these vertebrae. Why is the spinal cord important? Because the majority of the pathway that regulates the information exchange, the information movement from outside, from the skin, and also from the internal region of our body, are regulated by the spinal cord. So the information movement back and forth between the brain and our body is regulated by spinal cord. It also regulates the movement, the locomotion. If there are any kind of trauma or any kind of damage at the spinal cord level, as a consequence, we can have an impairment into the locomotion and impairment into the leg functions. Or in case of trauma, in the most severe condition, we can also have this as a consequence, we can also have the paralysis. So the loss of the ability to move our leg, or in some case the paralysis of all the down portion of our body, the loss of the voluntary control of muscles. So people that have any kind of severe impairment into the spinal cord do not have the ability to regulate the voluntary muscles. So even if you think that you can do it, your body does not respond to the voluntary control of muscles functions. So it is something important for the regulation of the information pathway and the movement, the locomotion. As we said, it is divided into these four regions, so the cervical region, the thoracic region, the green, the lumbar, and down here we have the sacral region. All of these regions have a kind of similar structure. If we look into the organization more in detail, you can say that the spinal cord is organized, is divided into gray and white matter. So, as we said, the gray matter is composed by cell body, so the nuclei that lie into the internal portion of the spinal cord, and then it is surrounded by white matter, so by myelinated axons. So, if you look more in detail into the composition of the gray matter, you can say that we can distinguish the dorsal and the ventral horn. So the upper portion that is composed by dorsal horn, and here the ventral horn. Of course, these structures have different functions. So from both of them, we have the formation of we have these two branches. That are part of the spinal nerve. So we can see the origin of both the dorsal and the ventral root. And from these structures, we have the information movement. So from the dorsal root structure, from the dorsal root ganglia, we have the information that move from the periphery, the peripheral nervous system to the central nervous system, using the efferent division that originates from the dorsal horn. From the ventral horn, we have the efferent division, so the motor information that regulates muscles, that regulate skeletal muscles functions or glands in some conditions. So basically the structure is more or less the same, two different horns, but from each one of these horns originate cells with different functions. So sensory neurons from the dorsal horn that carry information to the central nervous system, and efferent neurons that carry information from the brain, from the nervous system, and regulate skeletal muscle functions. If you look, so this is a representation. So you have the somatic sensory neurons that basically work into the dorsal horn, and the motor nuclei that regulates the efferent functions from the ventral horn. So the information are able to move back and forth from the spinal cord to the brain using these two different structures, the dorsal and the ventral horn, from which originate dorsal and ventral root ganglia. So this is just one again to summarize the organization of the spinal cord. So the sensory fibers from the dorsal roots, we are here, the dorsal root ganglia that originates from the dorsal horn that send information to the brain for the elaboration into the integrating centers, and then down the ventral horn. It contains the nuclei of that motor neurons that send afferent information, so that regulates the skeletal muscles. So this is, in general, the organization of the spinal cord. For what concerns the gray matter. So the gray matter, that is this internal portion that has this particular shape, but it is usually called the butterfly shape. But all this gray matter is basically surrounded by white matter. So it is surrounded by axons, by myelinated structures. We can distinguish, even in this portion, even into the white matter, the ascending tract, so that kind of sensory neurons that send information to the central nervous system, that are the majority basically located here in the top portion, and in some portion that surround basically the the gray matter. And then you also have the descending tract, so that kind of afferent division that regulates the skeletal muscle, that commands to motor neurons. So information that comes from the brain. And this the localization of this myelinated structure is in here. So you can see that even into the white matter, we have a division for what concerns the ascending and what concerns the descending tract. So the information, sensory and afferent division, are distinguished by themselves, and it's the same we observed for the gray matter, the dorsal and the ventral horn. So we can see both white and gray matter into the spinal cord. An important function of the spinal cord is the spinal reflex. So what means? We mentioned several times the integrating center that is located in the brain, in the cortex. That is the center for the central nervous system that elaborates the information, that processes the information and sends the output. For what concerns the spinal reflex, this information can be elaborated by the spinal cord, in the spinal cord by itself, without the brain, without the input, we don't need that the input arrive into the brain, and so the output comes from integrating center in the brain. In this case, everything takes place into the spinal cord. So you have the stimulus, the information that moves through the afferent division, and they are elaborated into the spinal cord. And it's the spinal cord by itself that, thanks to the elaboration of this information, send the output, and so we have the response that comes from the stimulus. In this case, we do not have the involvement of the brain. Of course, the information can also be sent to the brain, but for what concerns some kind of simple reflex, simple spinal reflex, this information can be elaborated in the spinal cord by itself. The spinal cord is able to elaborate these simple stimuli. Of course, from the interneurons, the information can be sent to the brain, and using the afferent division, the information, the output arrives into the ventral horn, and so we have the regulation of the response. But for what concerns the spinal reflex, everything is elaborated into the spinal cord. This is kind of an important function for the spinal cord, for that kind of stimuli that are simple and require a really immediate response. In this case, of course, the response is quicker, because we don't need to send the information to the brain, but everything happens into the spinal cord. So these are the main function of the spinal cord. This is, it's just an overview for what concerns the spinal cord. We're not going to go deeper into the detail of the spinal cord function. Just keep in mind that is the main information center. So using the spinal cord, information are able to be sent back and forth brain and the skin and our body. It is important in the regulation of locomotion, in the regulation of skeletal muscle function, and it is composed, as we're going to see for the brain, by both white matter and gray matter that regulate its functions. So we have the ascending and the division that regulates the movement of information to the brain, and the efferent division that regulates the output that comes from the brain to downstream organs, regulating the skeletal muscle functions. So this is what you should know about the spinal cord. Yeah, we can talk about the brain next time. We are going to see this other structure of the nervous system, of the central nervous system, that is the brain. We're going to go through the main structure, the main anatomical structure of the brain and their functions, and how brain and spinal cord are able to integrate their function to elaborate the response. But we can leave it for next time. I think it's enough for today. Guys, do you have any question? Ciao ragazzi.